Slide 1, nervous system. You have nerves throughout your entire body. Nerves send electrical signals that help you feel sensation and move your muscles. Nerves also control bodily functions like digesting food and maintaining your heart rate. Nerves are the foundational parts of your nervous system. Your peripheral nervous system, the nerves spread out from the spinal cord to different parts of your body, including your internal organs, collect information in the environment like touch, pain, breathing, and send it to the brain. Messages are really just electrical signals. The nervous system uses low voltage, tiny electrical signals to send messages. When you touch something hot, for example, your nerves in your hand send a small voltage electrical signal to the brain. Your brain processes this and sends the message right back, telling the muscles in your hand to move away quickly. So you can say that nerves are like cables that send electrical impulses between your brain and the rest of your body. Slide 2. Notice these two neurons. On one end are dendrites, which are highly branched structures resembling branches on a tree. The ends of all dendrites can initiate a nerve impulse. An impulse goes to the cell body and then gets transported to the axon. The axons can be very short, 1 centimeter or less, or they can be quite long, such as 3 feet. Most of the axon has a myelin sheath for protection. Myelin sheaths are made up of fatty substance. Along the axon are gaps in the myelin sheath called the nodes of Ranvier. You can think of the nodes of Ranvier as myelin sheath gaps that are insulated and have many ion channels in them. Although the axon to participate in the exchange of ions is required to generate nerve impulse. A nerve impulse is also called an action potential. These axons send a signal on to another series of tree-like branches called axon terminals. So a nerve impulse goes from the dendrites to the cell body to the axon terminals. Nerve impulses are made possible by ion gate channels. Slide 3. Ion nerve impulses are known as action potentials. Action potentials are tiny subunits of very low voltage electricity, which is crucial for rapidly transmitting signals throughout the cell's axons and the entire nervous system. These action potentials allow neurons or nerve cells to communicate with each other and with other cells in the body. These action potentials occur when the cell's membrane electrical potential or voltage of a neuron rapidly changes. They easily polarize and then depolarize due to the opening and closing of gated ion channels in the neuron's cell membrane. Note that these ion channels are tiny proteins embedded in the cell membrane of neurons or nerve cells. Ion channels open and close, allowing sodium and potassium and chloride ions to cross the cell membrane and move inside and outside of the cell. Slide 4. These are pictures of gated ion channels in the cell membranes of a neuron. Gated ion channels of sodium, potassium, and chloride can be open or not. Gated ion channels selectively allow certain ions, such as sodium, potassium, and chlorine, to flow inside and outside of the cell through the neuron's cell membrane. This allows an electrical potential to build up on either side of the cell membrane. When a neuron receives a sufficient stimulus, voltage-gated sodium ion channels open, causing a rapid influx of sodium ions which polarizes the membrane and generates an electrical signal. The slide shows these gated ion channels on the physical boundaries of the cell membrane. Gated ion channels are specialized proteins found in cell membrane of neurons that regulate this flow of ions across the cell membrane. These channels are essential for transmitting tiny electrical signals, which are known as action potentials. Signals transmitted from a dendrite to an axon are action potentials. The sodium and potassium pump. In cells, that refers to the protein embedded in the cell's membrane that actively transports sodium ions out of the cell and potassium ions into the cell. This maintains a crucial concentration gradient across the membrane, which is essential for nerve impulses to be transmitted and for muscle contraction. It uses energy from ATP to perform this transport against the concentration gradients of both ions. Slide 5 shows this action potential. Action potentials are nerve signals. Neurons generate and conduct these signals along their axons in order to transmit them to targeted tissues. Upon stimulation, they will either reach a small threshold or to fire, or they will be inhibited or modulated in some way. The ion channels are opening and closing and even using ATP for energy. A voltage potential gradient is found on the outside of the cell relative to the inside of the cells, awaiting to reach a small threshold in order for the action potential to fire. Slides 6, 7, 8, and 9. For these 4 slides, look at the purple circle in the graphs in the upper right. This is the graph of the action potential. The x-axis is time in milliseconds and the y-axis is electric potential or voltage in millivolts. Slide 6, the start of an action potential. It shows the uptake of sodium ions, but what causes an action potential? From an electrical aspect, it is caused by a stimulus with a certain value expressed in millivolts. Not all stimuli can cause an action potential. Adequate stimulus must have sufficient electrical value, which will reduce the negativity of the nerve cell to the threshold of the action potential. Sub-threshold stimuli cannot cause an action potential. When the threshold stimulus is of enough energy or potential, then it produces an action potential or nerve impulse. This is an all or none principle. Once the threshold potential is reached, an action potential will fire, and the action potential always fires at full strength, regardless of the stimulus intensity. An action potential consists of several phases, including depolarization and repolarization. Again, note the purple circle in the upper right-hand corner. Slide 10. This is the generic graph of an action potential. So to recap, the action potential is generated when a stimulus changes the membrane potential to the values of the threshold potential. The threshold potential is usually around -50 to -55 millivolts. It's important to know that the action potential behaves in the all or none law. This means any sub-threshold stimulus will cause nothing, while a threshold and a super-threshold stimulus produce a full response to excite the cell. An action potential is not different on whether it's caused by a threshold or a stronger superpotential threshold. The length and amplitude of an action potential are always the same. However, increasing the stimulus strength causes an increase in the frequency of action potentials. An action potential propagates along the nerve fiber without decreasing or weakening of amplitude and length. In addition, after the one action potential is generated, neurons revert to their initial stage. There is a refractory period of time in which they cannot generate another action potential. At the end, the cell then reverts to a -70 millivolt resting potential. Please note that the time involved is measured in milliseconds. Slide 11 shows the explanation of this action potential in a bit more detail. Again, the x-axis is time in milliseconds and the y-axis is in millivolts. Action potentials are quick and low voltage. The slide shows the threshold line for the action potential to take place. It also shows a failed initiation of an action potential, the refractory period, and then the neuron is back to the resting state. The entire process should take about 4 milliseconds. Slide 7. Continuation of an action potential. Again, note the purple circle in the upper right-hand corner. The action potential has gone past the point where the nerve impulse has been triggered. The sodium channels are now closed and the potassium channels are open, starting the repolarization process. Repolarization is a potassium efflux and is now at a brief hyperpolarization phase where the membrane potential dips slightly below the resting potential before returning to normal. Slide 8 is the end of the action potential. The purple graph again in the upper right hand corner shows how the resting phase is now restored to await another firing of the next action potential. The potassium ion channels are about to be closed. Let me stress again, this electrical potential or action potential is expressed in units of millivolts. Slide 9. Total completion of action potential. The potassium gates are now closed. So let's toggle back from slides 6 to 7 to 8 and then to 9. Please note the purple circle in the upper right hand corner and see how the action potentially actually plays out. Slide 12. This shows the action potential with the sodium and potassium ion gates superimposed upon it. Please note that when the sodium ion channel gates open and fully open and then close, and please note when the potassium ion channel gates open and close. This graph should mirror the slides 6, 7, 8, and 9 in the purple circle, denoting the stages of the action potential. Slide 13, neurons. Notice these two neurons. On one end are dendrites, which are highly branched structures resembling branches on a tree. The end of all dendrites can initiate a nerve impulse. An impulse goes from the cell body and then gets transported to the axon. The axon transmits the nerve impulse, and that's also called an action potential. It's transmitted to a series of tree-like branches called axon terminals. So a nerve impulse goes from the dendrites to the cell body to the axon terminals, which end at the synapse. A synapse is a gap between the axon terminals and the next dendrite. The synapse is the space between the two nerve cells where many very unique cellular activities take place. Slide 14. This shows the end of one presynaptic neuron and the beginning of the next or postsynaptic neuron. Please note that the gap between these two neurons or nerve cells. There is not an action potential or voltage gradient, but rather a release of chemicals from the presynaptic axon and a response on the postsynaptic neuron. The space in between the two neurons is called the synapse. This slide shows that the presynaptic neuron releases chemicals into the synapse where they are received by the postsynaptic neuron. Slide 15. When a nerve sends an electrical impulse, the signal travels down the axon, the wiring connection of the nerve. Note the red arrow. The message converts to a chemical signal at the end of the nerve cell, note the red circle, and at the end of the axon does not physically touch the dendrite of the next nerve cell. There is a space. There is a chemical release at the end of the axon of the first molecules. They release chemicals called neurotransmitters into the space that bridges the two cells. The space between one neuron and the other neuron are called synapses, and electrical signals travel down the length of the next neuron. Note the blue arrow. The synapse with the neurotransmitters are one of the utmost importance. Types of neurotransmitters and the actual chemical and electrical charges will be further explored. Slide 16. A synapse is a specialized connection between neurons that allow them to communicate with each other, and the synaptic cleft is the microscopic gap between the neurons where this communication takes place. The structure of the synaptic gap is between the axon terminal of one neuron and the dendrite of the next neuron. The synaptic cleft is typically 20 to 40 nanometers wide, which is much smaller than the width of a human hair. When a nerve signal reaches the presynaptic neuron, it causes the release of neurotransmitters into the synaptic cleft. These neurotransmitters then bind to the receptors in the postsynaptic neuron. Neurons talk to each other across synapses using chemical nerve signals. Synaptic vessels are little bags of neurotransmitters that are waiting at the end of an axon. Calcium ions activate these small vesicles of neurotransmitters via exocytosis. Then the neural transmission will actually occur when the neurochemicals are released into the synaptic gap in the synaptic cleft. Some of the neurotransmitters will bind or dock themselves to the receptors on the postsynaptic neuron. Some neurotransmitters are washed away, but some will recycle themselves. Neurotransmitters will go back into the axon of the nerve cell they just left. So some of the neurotransmitters will get reabsorbed by the original neuron. When you reabsorb these neurotransmitters, this is officially called reuptake. If the signal is repeated over and over again, the message gets stronger and is more likely to be transmitted, so the next message is even more likely to get through. In this way, neurons can pass information on with their important messages. Slide 18. There are at least 100 different types of neurotransmitters. They can be grouped into a few types based on their chemical nature. Some of the most common neurotransmitters include acetylcholine, dopamine, serotonin, glutamate, norepinephrine, endorphins, and GABA, gamma-aminobutyric acid. They are primarily found in the brain and central nervous system, where they are released from neurons to transmit signals to other cells. Acetylcholine is also found at the many neuromuscular junctions, which connects the nerves to muscles. Neurotransmitters are usually synthesized in the neurons that they are located in, and they are derived from precursor molecules. General classes of neurotransmitter include amino acids, monoamines, and peptides. Monoamines are synthesized by altering a single amino acid. As I've stated, communication is the interaction between neurons and the chemicals with the neuron as it fires and passes information. However, different neurotransmitters have different effect on the postsynaptic neurons. Some neurons will have an excitatory effect as it increases the likelihood that the neurons will fire and pass on the electric messages, and some neurotransmitters are inhibitory to the postsynaptic neuron. This decreases the likelihood of postsynaptic neurons to fire. Excitatory and inhibitory influences are the sum or the total of the two opposing influence. There are also modulatory neurotransmitters that influence other chemical messengers. They tweak or adjust how cells communicate with the synapse. They also affect a large number of neurons at the same time. Slide 19. This slide graphically depicts these two opposing neurotransmitters. Glutamate, this is the most common excitatory neurotransmitter of your nervous system. It is most abundant neurotransmitter in your brain. It plays a key role in cognitive functions like thinking, learning, memory, and imbalances in the glutamate levels are associated with Alzheimer's disease, dementia, Parkinson's disease, and seizures. Gamma-aminobutyric acid, or GABA, is the most common inhibitory neurotransmitter of your nervous system, particularly in your brain. It regulates brain activity to prevent problems in the areas of anxiety, irritability, concentration, sleep, seizures, and depression. Glycine. Glycine is the most common inhibitory neurotransmitter in your spinal cord. Glycine is involved in controlling hearing, processing, pain transmission, and metabolism. Slide 20. Here is a list of neurotransmitters that are desired neurotransmitters. The normal levels of these neurotransmitters are great. Dopamine, also known as the rewarding chemical that is released as a reward when people have a pleasurable experience. Too high a dopamine levels causes euphoria, poor impulse control, aggression, and hallucinations. Too low of levels of dopamine include unmotivated, unhappy, memory lost, and mood swings. But the right amount is desired, gives these good characteristics. The next one is serotonin, which is a mood booster stabilizer, and normal levels are great for people to be generally happier. However, too high of serotonin causes agitation, restlessness, high temperature, muscle spasm, and seizure. And too low of levels of serotonin can cause phobias, depression, anxiety, and even suicide ideation. Again, the right level of serotonin will give you the right mood. Endorphins is the third one. They are natural painkillers and stress relievers and control your well-being and anxiety. Too high levels of endorphins causes disorders like obsessive compulsive behaviors or even self-harm. Too low of levels of endorphins give you increased aches and pains and depression and anxiety and mood swings. And the last is oxytocin. It is known as the love hormone, but it really increases positive social interaction and reduces stress. High levels of oxytocin causes reduced empathy, difficulty in forming and maintaining relationships, a lack of happiness, difficult feelings of affection, and increased anxiety. And low levels cause irritability, disturbed sleep, and are linked to the autism spectrum disorder and depression. Again, this is the love hormone, and it promotes positive social interaction in the right amount. Slide 21. This depicts the difference in neurotransmitters in the synapse of a normal person and someone who is experiencing depression. Note that the amount of neurotransmitters and the amount of neural acceptors in the postsynaptic side. Slide 22. Depression is a common mental health condition that causes a persistent feeling of sadness and changes in how you think, sleep, and eat and act. There are several different types, but depression is treatable, usually with talk therapy or medications or both. There are several chemical treatments for depression and other mood disorders, including SSRIs. They are the very common selective serotonin reuptake inhibitors, and it's the most common prescribed antidepressants. SSRIs are often the go-to treatment for depression. They increase serotonin levels in the brain by blocking the reabsorption of the serotonin by neurons in the reuptake process of nerve transmission. Some examples of SSRIs are Prozac, Zoloft, Paxil, Celexa, and another group of chemicals are the serotonin norepinephrine reuptake inhibitors, or SNRIs. Similar to the SSRIs, the SNRIs increase serotonin and norepinephrine levels in the brain. Some examples are Cymbalta, Prestique, and Fatisma. Mood stabilizers. The mood stabilizers are used to treat bipolar disorder and other conditions that cause mood swings. Mood stabilizers reduce abnormal brain activity. Lithium and anticonvulsant drugs are some examples of mood stabilizers. I hope you've enjoyed and learned from this presentation on ion channels and neurotransmitters by Richard Pronchi, Grand Canyon University. Thank you.